Thank you for the invitation to come here. I hope to enjoy Barcelona. It looks like a great city. From my hotel room, I can look at the water on one side and turn around and look on the mountains at the other, and so that's absolutely fabulous. So I'm going to quickly move into this lecture since I have less time than I thought. So this is an overview of peroxisomal disorders diagnosis and management. And my colleague Hans Waterham will be presenting the complementary lecture on the need for continued need for metabolic assays and the unusual phenotypes we've diagnosed that way. So I have no disclosures. The learning objectives in this lecture will be to develop a clinical, biochemical, and molecular diagnostic approach, to review some of the current management approaches, and then appreciate how scientists, physicians, and family groups all interact to make a difference in rare disease, and everyone is a key player, which I'm sure you all know. So this is just a slide of what peroxisomes look like. So these are skin fibroblasts from a control individual that have been fixed and permeabilized and stained with an antibody that either recognizes peroxisomal membrane proteins or the matrix enzymes inside. And what you can see are hundreds of these spherical single membrane bound organelles that are present in all eukaryotes. They're small. They contain more than 50 enzymes. And what's unique about them is their versatility and that they can really proliferate and regulate their enzyme composition according to metabolic and environmental cues. And so there are multiple enzymatic pathways in the peroxisome. This slide is just a list of some of the more well-known pathways. And so peroxisomes are known for their multiple enzymes that are involved in fatty acid oxidation. And these are long-chain fatty acids greater than C22 in length. And they're a combination of saturated, unsaturated, dicarboxylic fatty acids, and as well methyl branch fatty acids like phytanic and pristanic acids. And in the oxidation of these acids, actually, there are products that are formed as well. So there's the synthesis of docosahexaenoic acid, an important omega-3 polyunsaturated fatty acid, also called DHA, that is generated in the peroxisome from one step of beta oxidation from its C24 precursor to the C22 final product. And then bile acids are synthesized the same way, so intermediary bile acids of C27 in length are oxidized in the peroxisome to the final mature bile acid, that's the C24 cholic and chenodeoxycholic acids. So peroxisomes are also involved in amino acid oxidation, such as piperacolic acid, which is in the lysine catabolic pathway, and also the conversion of glyoxylate to glycine so that oxalate doesn't build up and cause renal stones. And peroxisomes are also involved in the synthesis of plasmalogens, which are specialized membrane glycerophospholipids that I will talk about in a bit. And in these oxidative reactions, peroxisomes produce hydrogen peroxide, which is how they got their name. And hydrogen peroxide needs to be consumed, and so peroxisomes have the enzyme catalase as well as other antioxidant enzymes to help with the reactive oxygen species balance in the peroxisome and in the cell. So just to go over the enzymes involved in the beta oxidation pathways. And so the reactions are the activation of the very long-chain fatty acid to its CoA derivative. And then there's the hydration and dehydration reactions, which are carried out by a bifunctional protein called D-bifunctional protein. And then there's a thiolysis where the N2 carbon unit is removed, and then we turn around for another step of beta oxidation until we get to the final product. And what this also shows you is that the different fatty acids that I mentioned before, the straight chain, saturated and unsaturated, the branch chains, this is the DHA and the bile acids have some overlapping, but also some different enzymes. So the acyl-CoA oxidase 2 is responsible for oxidation of the branch chain, where acyl-CoA oxidase 1 for the others. D-bifunctional protein pretty much is involved in all of the oxidative steps. And then we have two enzymes involved in the thiolase. And what this also shows you is that if you have what helps us with our diagnosis when we look at metabolites is that if we have a defect in ACOX2, we're going to expect different types of very long-chain fatty acids to accumulate than if we had a defect in ACOX1. And similarly, because of enzyme redundancy, if we have a defect in this thiolase, ACAA1, we actually don't see any clinical defect because there's a second thiolase, SCPX, that can take over in all these reactions. So the pathways are helpful to know and can help us tease out the underlying defect. And then this shows you that in order to get into the beta oxidation pathway, some of the fatty acids actually have to go through a preliminary oxidative steps. So for phytanic acid, there needs to be an alpha oxidation step in which the product is pristanic acid. And pristanic acid comes in two enantiomers, which I've written down here are non-superimposable stereoisomers in R and S form. And there's a racemase, alpha-methyl-CoA racemase, that converts this all to the S form, which can then enter the beta oxidation pathway. And for bile acids, similarly, all bile acids are in the R form, and they have to be converted to the S form too. So again, if we have a defect in the alpha-methyl-CoA racemase, we could predict that both of these metabolites would be elevated, but if it's alpha oxidation, only the phytanic acid would be elevated. So, and this is just a quick review of plasmalogen synthesis, which is initiated in the peroxisome. And so it starts with this substrate, dihydroxyacetone phosphate. And then fatty acid is added to the C1 position up here by dihydroxyacetone phosphate acyl transferase, or DAPT. And then the fatty acid is exchanged for a fatty alcohol by dihydroxyacetone phosphate acyl synthase, or ADAPS. And the fatty alcohol is obtained from the enzyme FAR1, which is located on the peroxisomal membrane. And then this product here, which is called a 1-alkyl dihydroxyacetone phosphate, is actually sent into the endoplasmic reticulum for a number of other steps to make a mature plasmalogen. So the top of ether-linked alcohol is converted to a vinyl ether alcohol with this double bond. Another fatty acid is added to the C2 position, and then we have the phosphatidyl ethanolamine, or choline, as the phosphate head for this membrane glycerophospholipid. So all of these enzymes are targeted to the peroxisome matrix by virtue of their containing peroxisome targeting signals. And there's two types: peroxisome targeting signal 1, which is a C-terminal serine, lysine, leucine, or consensus variant thereof. And this is by far the majority of peroxisomal matrix enzymes carry the PTS1 signal, and the receptor is PEX5. A few peroxisomes contain a type 2 targeting signal, which is a more complicated nanopeptide, which has actually recently been shown to be a structural motif that fits nicely into a groove in PEX7. And there are 3 well-characterized PTS2 enzymes. There may well indeed be more that are tissue-specific or timing-specific. And this includes that thiolase, which we know is redundant, and also the phynyl-CoA hydroxylase to alpha-oxidize phytanic acid. And this enzyme I just spoke to you about, which is important in plasmalogen synthesis. And as I mentioned, there's an expanding number of matrix enzymes, and this comes from both bioinformatics that looks for these targeting signals, but also tissue-specific proteomics. And then there's also examples of matrix enzymes that don't have a targeting signal, but actually hitch ride onto the back of the PTS1 and PTS2 enzymes, which is called piggybacking. And so peroxisome biogenesis, or assembly, is 2 processes, which is matrix enzyme import and formation of new peroxisomes. And this is accomplished by a coordinated set of PEX proteins, or peroxins. And just to go over this really briefly, the first step is import, where the PEX5 and PEX7 receptor bind their cognate ligands, and this complex is translocated to the peroxisomal membrane, where 5 and 7 dock at peroxins 13 and 14. The ligands are distributed or translocated into the peroxisome matrix. And then PEX5 is ubiquinated by this set of proteins here, PEX2, 10, and 12. And this complex at the end, PEX 1, 6, and 26, uses the energy of ATP hydrolysis to actually move and pull PEX 5 out of the membrane by its ubiquitin handle, and out pops PEX 7 also, and then the receptors are recycled for another round. And in peroxisome matrix protein import, peroxisomes can import fully folded proteins, and so this is a dynamic pore here that can open very widely. It is thought. It's also important to know that some of these enzymes are shared between different cell compartments, and that there's a large amount of organelle crosstalk that we're just beginning to learn more about. And then the other process is the formation of new peroxisome membranes. And this is an old slide, but it gives you sort of the basic idea here, which is that the peroxisome membranes come from ER membranes. There are 3 peroxins, PEX 3, 16, and 19, that are involved in recognizing the peroxisomal membrane proteins, bringing them to the peroxisome membrane so that a mature peroxisome can be formed that can then import matrix enzymes, and then this organelle can be divided by fission using the PEX 11 proteins. And the fission machinery is also shared by some mitochondrial proteins that are involved in mitochondrial fission. And in actuality, what happens in the cell is that a combination of the de novo peroxisome formation and division by fission occurs, so that you have division by fission, and that would make a smaller peroxisome, and new membrane material will be constantly added from the ER. So the 2 processes occur together. And biallelic mutations in any of 13 of these PEX genes would cause peroxisome biogenesis disorders in the Zellweger spectrum. And as you can just appreciate, this is going to cause reduced peroxisome numbers, failure to import matrix enzymes, and we're going to end up with multiple enzyme deficiencies, which is going to result in the accumulation of toxic metabolites that are not getting degraded, as well as deficiency of compounds that are not being synthesized. And that end result is what causes these developmental and multisystem progressive disorders. And then biallelic mutations in PEX 7 cause the peroxisome biogenesis disorder RCTP1. And in this disorder, there are normal peroxisome membranes. There's failure to import the PTS2 enzymes. The disease is due to a defect in plasmalogen deficiency because of the failure to import the PTS2 targeted ADAPS enzyme, and this also results in complex developmental and multisystem progressive disorders. And so the historical classification of peroxisome biogenesis disorders was that of multiple malformation syndromes. So you had Zellweger syndrome, which was also called Cerebro-Hepato-Renal syndrome, and rhizomelic chondrodysplasia punctata. And these disorders were also classified based on neurological dysfunction. So you had neonatal adrenal leukodystrophy, or NALD, and they might have also been classified according to their liver disease and failure to thrive in older kids, and so they were classified as infantile Refsum's disease. But now that we know that they're all due to a peroxisome etiology, the current classification of these disorders is that of peroxisome biogenesis disorders due to PEX gene defects, which give multiple enzyme deficiencies, and that would be Zellweger spectrum disorder and rhizomelic chondrodysplasia punctata. And we also have a group of single enzyme or protein deficiencies, the most common of which is X-linked adrenal leukodystrophy, which is due to a transporter on the peroxisome membrane for very long-chain fatty acids and is actually not a PEX gene defect. And the current favored term usage is Zellweger spectrum, severe, intermediate, mild. So we're no longer using Zellweger syndrome, neonatal adrenal leukodystrophy, and infantile Refsum's disease to describe those same categories. And RCDP is also a spectrum of severe, intermediate, and mild. And we're using the term spectrum because we feel it's more inclusive of the common peroxisome etiology. Patients are all individuals on the spectrum. There's a lot of overlap, and because of all of the expanded and new phenotypes being reported that don't fit into any of those prior diagnostic classifications. And so in order to recognize Zellweger spectrum disorder in the clinic, we do go back to that original description, Cerebro-Hepato-Renal by Hans Zellweger, and we add adrenal. So if you keep in mind the systems involved in these disorders are Cerebro-Hepato-Renal-Adrenal, you will probably pick up many of the children and adults. And so what I've divided these classifications by the blue star are the phenotypes that are caused by in utero defects or being malformations. And so the peroxisome disease is so severe that there's an effect in utero. And the other phenotypes are more progressive phenotypes over time due to peroxisome dysfunction. So for the brain, there's a characteristic dysmorphic facies. There's also neuronal migration defects in the severe disease, and leukodystrophy and atrophy can develop as a progressive condition. There's impaired psychomotor development, hypotonia, areflexia, and seizures. You can have congenital glaucoma in the eye, optic nerve hypoplasia. Cataracts actually can occur at any time, so they can also be a manifestation of a progressive disease. Retinal dystrophy, which leads to blindness. And then sensory neural hearing loss, which leads to deafness. And in the liver, there's hepatomegaly, cholestasis, cirrhosis, due to elevated bile acid intermediates, which are toxic to the liver. And the liver disease also includes elevated INR due to vitamin K deficiency because of the bile acid deficiency and the inability to absorb fat-soluble vitamins. There's also multiple cortical renal cysts, hydronephrosis, and patients can develop hyperoxaluria and renal stones. Adrenal insufficiency can develop, and then the other manifestations besides in the Cerebro-Hepato-Renal-Adrenal axis would be chondrodysplasia punctata in the hips and knees, which would be a developmental malformation, osteopenia and enamel hypoplasia of the secondary teeth, which are progressive conditions. And so just to recognize severe Zellweger spectrum or Zellweger syndrome, so these kids were recognized by their dysmorphic features and the other phenotypes. And you can see that this is a recognizable dysmorphic feature that when you see over and over again becomes much more familiar. And so there's hypertelorism, there's a broad forehead, there's a small midface and a upturned nose, a small chin, and depressed supraorbital ridges. And there's also redundant folds around the back of the neck and often a transverse palmar creases. So this little girl is jaundiced because of her liver disease, and they're all sort of really floppy, and they need feeding tubes. And they generally don't survive because of the neuronal migration defects. And so here's just a slide that illustrates that normally the neurons migrate from the outer to the inner layers, and so there's a failure of migration, and what that ends up causing is the gyral defects, polymicrogyria, pachygyria, and heterotopias. And there's a classical MRI in the severe form in which you have polymicrogyria in the sylvian fissure and pachygyria up in the frontal lobes. So intermediate severity was originally classified as neonatal adrenal leukodystrophy, so you assume the children develop a leukodystrophy. Not all of the intermediate patients develop a leukodystrophy, and the course of the leukodystrophy can be very different. And so this little boy developed a leukodystrophy and died soon afterwards. However, this little girl developed a leukodystrophy which arrested, and she has now regained functions and is 10 years old. And this woman was living in a group home and had blindness and deafness, and she had some behavioral changes that led to the diagnosis of a leukodystrophy at 42 years of age. And then these are kids showing intermediate and milder severity, and there's going to be a whole overlapping picture of intellectual deficiency with some children actually being in normal levels in school, hypotonia, hearing, and vision loss. And so I think in the milder cases, you just have less of these manifestations, and they're milder, and in the more intermediate cases, you have more of this expanded phenotype, and it's a faster progression. And so this would be another example of milder Zellweger spectrum disorder. This is a 32-year-old woman who was identified by whole exome sequencing. She was deaf and blind, and she had ameliogenesis imperfecta. She had a normal MRI, had no adrenal insufficiency, had no liver disease. She finished university and has a professional career. This was reported in 2011, and I think this patient and many others, as Hans will present, are showing to us that this spectrum is broad enough to include normal intellect. And then we also see variation within a SIBSHIP, so that this 18-year-old is the sister of that child with the leukodystrophy that arrested, and she has normal intellect and deafness and blindness. And she does have white matter changes on the MRI, but in this sense they were silent, and she's never had any clinical symptoms. And then there's another group of atypical presentations of Zellweger spectrum disorder, where instead of these sensory findings of hearing and vision loss that you've heard me talk about, hearing and vision remain intact, and in general, intellect remains intact, and the presentation is an ataxia and peripheral neuropathy, or various combinations. And here's a 5-year-old boy who presented with ataxia and areflexia and normal intellect. He was normal for other parameters, but he did have hematuria with calcium oxalate renal stones. And here he is at 5 years of age, and here he is at 13 years of age. And what this disorder was also severe in the sense that there was a rapid progression of the peripheral neuropathy leading to death. And so I think what I want to have you appreciate today is that in the severe versus milder Zellweger spectrum presentations, in the severe form, we can always pull those cases out because developmental malformations will be present, and those will include malformations in the brain, renal cysts, chondrodysplasia punctata, and those kids in general do not have a long lifespan and pass away before 1 year of age, mainly because of the defects in the neuronal architecture. And then there's intermediate milder forms where the developmental malformations are absent, and this is really a progressive disease of ongoing peroxisome dysfunction with hearing and visual loss, maybe a leukodystrophy, maybe adrenal insufficiency, maybe renal stones, maybe osteopenia. And then there can be a different presentation with ataxia and areflexia, reflecting the peripheral nervous system and cerebellar atrophy. So I'm not going to review these because of time, but this is just a nice little chart that we've put together that shows you all of the features and how the malformations pull out the severe cases, and then the other disorders that are progressive phenotypes over time really sort out the intermediate and milder patients, and that the intermediate patients tend to have more of these phenotypes. So this phenotypic correlation also correlates with peroxisome number and size and degree of protein import. And so we're back to skin cells here, and the red is skin cells stained for peroxisomal membrane protein, so you can see the number of peroxisomes and their size. And here we're staining for matrix enzymes inside, so you could see whether these peroxisomes are actually importing enzymes. And so you can see in the controls you have numerous small peroxisomes, and they're all importing matrix enzymes. In the severe disease, you see much fewer peroxisomes that are enlarged, and these have been called ghosts because in general they're not importing anything. And then in the milder cases, we see many more peroxisomes, but they're not normal size, they're enlarged, but many more of them are functional and importing. And then these phenotypes also correlate with biochemical severity to some extent. So again, our diagnostic markers are very long-chain fatty acids, which is the beta oxidation, phytanic acid, which reflects alpha oxidation, and then plasmalogen synthesis. And you can see that in the more severe forms, we have more severe abnormalities. And in the mildest form, we have the milder abnormalities. And as Hans will be telling you, in some of the mildest cases, we don't have any abnormalities, so we have to look at other peroxisome tests. So these phenotypes also correlate to some extent with PEX gene mutations. And so, as I mentioned before, the 13 PEX genes that cause Zellweger spectrum disorder and defects in PEX1 cause RCDP spectrum. There are two alleles that are common due to a founder effect in Northern European descent. And one of these is a missense allele that has residual function in homozygosity and predicts a milder phenotype. And then there is a frame shift allele in which homozygosity predicts a severe phenotype. And then the combination actually predicts more of an intermediate phenotype. And then we know that missense alleles in a certain domain of PEX 2, 10, and 12 actually result in this atypical phenotype with ataxia and peripheral neuropathy, as well as missense alleles in PEX 16. So we have, I'm just going to skip RCDP in the essence of time. And move on to say that 15% of patients with PBD phenotypes will actually have a single enzyme deficiency and not a PEX gene deficiency. And this includes defects in beta oxidation that look like Zellweger spectrum, so defects in acyl-CoA oxidase, the D bifunctional protein that I mentioned, and also the racemase. So this tells us that it may be that of all of the enzyme defects in peroxisome biogenesis disorder, it may be the beta oxidation defects that are most responsible for the ZSD phenotype. And RCDP can also be caused by defects in Plasmalogen biosynthesis. There's an RCDP2 due to defects in DAPIT, RCDP3 to ADAPs, and then these new defects that Hans will tell us about where the PEX5 binding site to PEX7 is mutated, and so you get an RCDP5, and also defects in fatty alcohol reductase. And so, of course, this tells us, like I mentioned before, that it is the Plasmalogen biosynthesis defects themselves that cause RCDP. These would be the three major pathways in diagnosing these disorders, which I had gone over: alpha oxidation, beta oxidation, and lipid biosynthesis. And then metabolite testing needs to be confirmed by establishing a fibroblast culture where we can look at many other functions of the peroxisome, including, as you can see, what the peroxisomes look like and how they divide and how they import. And of course, the confirmation of testing is also done by DNA. So for the future, what is coming up, we have questions that my patients ask me all the time. So can you improve diagnosis? Can you improve prognosis? And can you improve treatment? So for diagnostics, we have there's been an enormous improvement over the last couple of years, and you know that we have next generation sequencing panels for PEX genes. And using these panels, there was a PEX6 founder mutation was identified in French Canadians with a huge increase in frequency from the 1 in 50,000 that I showed you earlier to 1 in 12,000. Whole exome sequencing has expanded the phenotypic spectrum of PBD. And now we have newborn screening being initiated now in the States for elevated very long-chain fatty acids. The primary target is X-linked adrenal leukodystrophy. The secondary target would, of course, be Zellweger spectrum disorder. And we're excited about this because it should tell us an accurate disease incidence, and it should allow earlier disease identification in asymptomatic patients. We need to improve prognostic information, and we have international longitudinal natural history studies going on for this at clinicaltrials.gov and OFFINET. You'll be able to find them. But as you can tell, we really need to understand what are the risk factors and the cause of these various phenotypes, like the leukodystrophy, the vision and hearing loss, the adrenal insufficiency, and renal stones. So if that little boy came up and asked me, would he develop a leukodystrophy, for example, I'm just being a little funny, but he's not going to ask me that, but I would have no risk factors that would predict it right now. So these are the things that we need to collect from the natural histories, make additional genotype-phenotype correlations, and then come out with biomarkers and accurate endpoints for interventional trials. And for supportive management, well, there's a lot to do. So we should not be telling our patients, you have Zellweger spectrum disorder and there's nothing we could do, or you have RCDP and there's nothing we can do. There's actually a lot you can do in supportive management. And so you can have, make sure that the child is getting regular vision, hearing, and neurological exams, for example. And for hearing, once hearing can no longer be amplified by hearing aids, these kids have received cochlear implants, at least in the United States, and they've done very well. You need to monitor liver, renal, and adrenal functions, as I have mentioned, and usually vitamin K is provided to prevent coagulopathy, adrenal replacement therapies when they're needed, and you want to be able to identify and prevent and/or prevent renal stones, which could lead to and have led to hypertension and renal failure. There's multiple dental interventions because of the ameliogenesis imperfecta with multiple cap teeth, pulled teeth, and different types of bonding, and physical and occupational therapies, for example. So there's also development of targeted therapies on the horizon, and so this would include therapies targeted to the metabolic defects or to the molecular defects. And so targeted to the metabolic defects, we can restrict phytonic acids, such as the diet that adult Refsum's disease patients get. We can supplement DHA and bile acids, because I mentioned before there's a deficiency of that, and plasmalogens. There are various case reports of this in the literature, more formal trials are needed in order to decide whether this is really helpful in changing the ultimate course of the disease. But this is particularly important since there is now a company that is making bile acids called Retrofin, and the bile acid is called Cobam. So we really need to be able to identify which individuals from the Zellweger spectrum disorder will respond to bile acids and will be helpful in changing the ultimate course of the disease. And then there's groups that have been working on drugs that enhance the oxidation, the omega oxidation of very long-chain fatty acids, which doesn't use the peroxisomal system. There have been cases of liver transplants that have rescued some of the biochemical abnormalities in the blood. And there are now current research going on drugs that can rescue the molecular defects. So the questions that we've been asking is how we can enhance the activity of a defective protein, how we can induce peroxisome proliferation, and how we can reduce peroxisome degradation. And we've been all particularly looking at this common allele because it's a missense allele with residual activity, it's partially degraded, it's temperature sensitive, and it displays this mosaicism in cell culture, which all make it actually a very druggable allele. And so we've been looking at various assays, and there's different assays that are in the community right now. But basically, the idea is to take a patient's cell line. In this case, we're expressing a green fluorescent protein with a PTS1 tag. No treatment at 37 degrees, it's cytosolic. If you bring the temperature down, you see that this mutation is temperature sensitive because at lower temperatures we recover some import. And if you bring the temperature to 37 degrees, but you add these nonspecific chemical chaperones like betaine and glycerol, you can also rescue import. So these are the kind of assays that are going on, looking for drugs that would recover peroxisome function in patient lines with certain mutations. And then there's also a bunch of model systems available in which we'd like to test these new therapies before we bring it to humans. And so really we've been calling this year the year of the peroxisome, and we hope to make a lot of advances that we can bring back to patient care. Thank you.
Thank you. I'm tempted to hop over Hans Walter and get you to do it one more time, because that was really excellent. Oh. So two things, as a clinician, we have some time for questions, so put your thinking caps on. Come back. Two things strike me as a clinician. One is that peroxisomes are sort of like, peroxisomal disease is sort of like, or Zellweger spectrum disease is sort of like mitochondrial disease. You've got to think about it all the time now. And the other thing is that it is tricky is the variation within a SIB ship, which can really sort of throw you for a loop.
I think so. And,
you need to go back to the microphone.
I think so. In the variation within the SIB chip, the way that we've been seeing it is that you see the older child usually who might be more severely affected because of those years that the peroxisome disorder has progressed, and then you see the younger kid who's doing so well. And so you don't know exactly which direction that younger child will go in. And then the example I gave was sort of the opposite, where the older girl had done well, and the family was never diagnosed until the younger girl presented with the leukodystrophy.
So scary. From a clinical point of view.
Yeah.
All right. Let's have some questions. She's come all the way from Canada. Okay. Please go to the microphone and identify yourself.
Jorge Hassin I'm from here, but I work in Gothenburg in Sweden. My question is, do you have NGS panel for PEX genes. If you have a suspicion of peroxisomal disorder, would you start with an NGS panel or would you do the biochemistry first?
Well, Hans,
Go to the microphone, Nancy.
Well, I think that would come from, if you ask a molecular person, they might give a different answer, but Hans will give you some more examples. We do believe that the biochemistry is really important. It can, as I tried to bring out in this lecture, it can point to you whether you might have a single enzyme defect or multiple enzymes. And it might be able to, if it's a single enzyme, then you might not have to do a gene panel, but just that enzyme. So I think it's useful to get at least some biochemistry before you move to, and decide that you do have a peroxisome disorder before you move to a gene panel. You could still go through a fibroblast cell line with confirmation, but but the idea is to get a molecular diagnosis for all of these patients. So then, of course, there's the harder patients where you do the metabolite testing and you don't find any abnormalities. And for that, you would have to go straight to a gene panel or to whole exome sequencing.
And what you do might depend a bit on your local circumstances and what's available and what things cost.
Yes.
Okay, I think there was another question. Please say who you are and where you're from.
Mrs. Wahl from Germany in Augsburg, genetic counselor. And I have a question whether the parameters, the very long fatty acids and the photonic acids can be found in blood as well or only in skin fibroblasts?
Can be found in in blood? Or?
The abnormalities, they can be found in blood or only in skin fibroblasts?
Yeah. So the metabolites are measured in blood and the enzymatic activity is measured in the skin fibroblasts.
Okay.
But we can also measure the metabolites in skin fibroblasts as well. For photonic acid, it's dietary only and it comes from fat sources of food that come from ruminant animals. So photonic acid comes from a specific set of foods. And newborns, since they're getting breast milk or formula, don't receive any photonic acid. So their photonic acid will never be elevated because it's not in their diet.
Thank you.
We have time. No question is too stupid. This is an educational session.
Dr. Ali Salim from Weill Cornell in Qatar. The features or the clinical finding of progressive peripheral neuropathy.
Wait a second. You're going to have to start over.
Okay, so the clinical finding of progressive peripheral neuropathy or cerebellar atrophy and ataxia, are they commonly seen among in the spectrum of your cohort of patients or rarely encountered or only seen at late stages? This particular feature of progressive peripheral neuropathy, cerebellar ataxia, and atrophy.
Right. So I think what I tried to show is that there is a group of patients now with certain defects in PEX 210 and 12 and PEX 16 that are showing that as the major presentation, and they don't have sensory deficits. So it's sort of different than your typical picture of Zellweger spectrum. But peripheral neuropathy by itself, we've seen that across the board, but not really as the presenting feature. So I wanted to sort of divide these two groups as sort of the typical presentations and this atypical presentation.
So it's a kind of atypical presentation?
Yes, it is. When it presents that way, it's an atypical presentation. When it's the major part of the disease, there's no vision or hearing loss, and that at the moment seems to be restricted to a set of certain mutations in certain genes showing a genotype-phenotype correlation.
Thank you.
Okay. You can tell they've been listening. Stand on your toes. This is Katarina Bjerger from Oslo, and she has a patient
who has, or siblings who have a PEX3 variant, and they have a mild Zellweger or intermediate, and they have abnormal peroxisomes. There's larger and fewer, but they have no metabolic changes. What do you think is the reason why we don't find any any biochemical changes?
Well, yeah, so I agree with that idea. So the more severe the patient, certainly in the intermediate category, we ought to be able to see those biochemical changes. And really where we have a tough time is the very milder cases. So I'm not sure. I would have to ask, what are you measuring? And that there's, there's your branch chain, so phytanic and pristanic acid, your straight chain, which is just your C26 to C22 ratio, your plasmalogen levels, your piperocalic acid levels, your bile acid levels.
We've had them in Hans Waterman's lab, and they've done everything, I think.
Well, we can talk about that afterwards. So that is very interesting. You would expect to see some abnormalities at least in a patient with an intermediate form.
Okay, thank you.
So you wouldn't have a situation like you can have with mitochondrial disease where the changes in the peroxisomes are secondary to something else?
Oh, but she said there was a PEX3 variant here.
Yeah, but if she hadn't said that, let's say that's a red herring and she's wrong about that.
That could be an alternative. So have you looked into a differential diagnosis?
Do you understand what I mean?
Yeah.
So that, because you see that sometimes in mitochondrial disease, right? That the mitochondria are down on their knees because of something somewhere else. So, okay, you guys talk afterwards. Unless, Hans, you want to say something?
That's also something you can see in Zellweger.
Oh, so,
Vice versa. Yeah.
okay, so Hans just said that you can see in Zellweger syndrome that the mitochondria don't look healthy.
And that was my example where the organelles interact.
Uh-huh. Okay. We can do another question. You got to use her while she's here. She's going back to Canada.
But I can still be reached.
I'm Judith Kerteszi from Hungary. I would like to ask if a child would be found early with this newborn screening, could we make this child healthy with our current knowledge?
No, there's no cure for these disorders, but as I mentioned before, there's a large community of people that are actively looking, I think, in very rational ways of finding more targeted therapies. But I think the newborn screening, remember, is being done as the primary target is X-linked adrenal leukodystrophy, in which there is a real intervention that can be done. So these kids are going to be picked up as a secondary target in that screening, and I think that there are advantages of picking up children that might be more intermediate or milder, and so the family doesn't have to go through that diagnostic odyssey. There'll be a chance of picking them up, and so that you can know what to monitor because you have a list of things that you now know you could do. You could probably, one of the problems we've been seeing is adrenal insufficiency is often not looked for, detected, or treated till very late. And so all of these things you can now move in doing them in a preventative and timely fashion to give that child the best quality of life.
So that's a good point, because one of the main premises of newborn screening is that you can fix what you find. Mm-hmm. And here you're finding something that you wouldn't look for as a byproduct of the screening. And then you have to, if you decide to do that, you're paying a price, in essence. Because, of course, but then on the other hand, you can argue these are recessive disorders.
There you go, yeah. You can also provide the parents with carrier testing and prenatal diagnosis in the next pregnancy before they have the second child.
Who may or may not be more severely affected. Okay, thank you very much, Nancy.